This segment of video or lecture will be on leukocytes, or the white blood cells. So let's talk about those, and again we'll be going through a day in the life of a leukocyte, where leukocytes come from as birth, then adolescence, structurally what is a white blood cell or leukocyte, and then into adulthood, what do they do, and then death, what happens, where do they go, and rate of turnover. So if we look at this graphic is what we saw at the beginning of lecture. So again, we've got our pluripotent stem cell and our red bone marrow, and this pluripotent stem cell goes through the process of hematopoiesis. This is true for all of the formed elements, and again, it takes place in the red bone marrow. Now, when we're talking about white blood cells here, we've got cytokine that can increase the number of progenitor cells. So those progenitor cells can then differentiate into a variety of different precursor cells. And there's two types of cytokines that are important for our white blood cells. There's colony-stimulating factors and there's interleukins. There's a video on colony-stimulating factors posted on Canvas, so make sure you go check that out when we're done with lecture. But each of these, so the pluripotent stem cell can differentiate into basically two types of cells, the myeloid stem cell or the lymphoid stem cell. And the myeloid stem cell then can differentiate into a couple, actually three different choices. The colony-forming unit, a granulocyte macrophage, and that'll differentiate into the myeloblast or the macroblast, which will then eventually differentiate into mature into a neutrophil and a monocyte, or a eosinophilic myeloblast or basophilic myeloblast, and those differentiate into the white blood cells you're familiar with, eosinophil and basophil. The pluripotent stem cell then differentiates directly into the lymphoid stem cell, and that can develop into one of these three white blood cells, the T lymphoblast, the B lymphoblast, and the NK lymphoblast. Those will then differentiate into the T lymphocyte or T cell, the B lymphoblast into the B lymphocyte or B cell, which could then convert into a plasma cell, and the NK lymphoblast will differentiate into the natural killer cell, which has the coolest name of all time. So that's essentially what it does. So let's look at kind of what the white blood cells are structurally, what they look like. So all of our blood cells kind of form in our red bone marrow. The white blood cells, however, have some maturity that they have to go through. So, well, white blood cells, many of them only live a few hours, for example, as phagocytic white blood cells. Some a few days, that's most of them, up to several months, or years, those are our lymphocytes. There's very few cells of blood, which you saw in lab. They're pretty hard to find. They can be higher if we're actively fighting an infection. On the opposite end, they can be lower as a result of radiation, shock, and certain chemotherapeutic agents that can reduce those numbers. At any given time, approximately 2% of the white blood cell population is in circulation. The vast majority are in skin, lungs, lymph nodes, and spleen, and they're kind of residents there. They kind of stay there. However, most need to be able to lose the bloodstream. Move into the bloodstream, and that's what we're looking at in this graphic here. This is called emigration. You'll see and hear the term diapetesis, but physiologists are now using the term emigration. Our white blood cells have adhesion molecules so that they can stick to the endothelium. So we've got our neutrophil here. It's floating in this capillary. You see this lovely capillary bed here. And it can stick to the endothelial cells. So these are selectins, they're a type of protein. And these green squares, these are integrins. These can stick to those selectins and then basically kind of wiggle their way through gaps in between the endothelial cells. What the heck is the point of these selectins? The endothelial cells display them in response to injury and inflammation. So these should not be here when we're healthy or when there's not anything exciting going on. These selectins are only present when we're fighting infection or there's inflammation or there's injury. Okay? So once these are displayed, then our white blood cells, in this case the neutrophil, can make its way into the bloodstream. Out of the bloodstream and into the tissues. Okay, so let's look at the different types of white blood cells. Hopefully this is a review from what you learned in lab. So we have our eosinophil. This is the most rare, least understood, 2 to 4% of the white blood cells. In some studies, it's released toxic compounds. They seem to be phagocytic. Work's still going on there. Basophils, this is the least common, contains heparin, histamine, and serotonin. Maybe that gives you an idea of what they might do. They intensify the inflammatory reaction. Imagine your itchy eyes and runny nose. They're involved in hypersensitivity or allergic reactions. So imagine somebody who's having anaphylactic shock. These guys play an active role in that reaction. We've got our neutrophils. These are 50 to 70% of our white blood cells. These are phagocytic. They especially like bacteria and fungi. They're generally the first to respond to tissue damage from bacteria. And they engulf the pathogen and then release a suite of chemicals. There's 3 different types to destroy the other pathogens. So let's talk about that. So one of the chemicals they can release are lysosomes. These are enzymes that will destroy certain bacteria. They also have strong oxidants. The peroxide anion, which is O2 negative, hydrogen peroxide, which is H2O2. Hydrogen peroxide, have you ever poured hydrogen peroxide on a cut? It's a really strong oxidant. Kills everything, including our own tissues. Hypochlorite anion, which is OCl minus. Essentially, these are free radicals, and they break cell walls, so they're very efficient at destroying bacteria. We also have, the neutrophils also have what are called defensins. These are proteins with a broad range of antibiotic activities. These are contained within vesicles, and these are pretty crazy. They form peptide spears that actually poke holes in the microbe membranes. Super cool. Imagine just molecular-sized spears that literally poke the hole in the bacteria so that they lyse. Fun stuff. All right, let's go on to the lymphocytes. These are about 20 to 30 of our white blood cell immune support. These ones, there we go. These ones are continually moving through lymphoid tissues, lymph, and blood. Generally, they only spend a few hours in the blood. However, they're spending most of their time in the tissues, the lymph tissues and the lymph. There's 3 types of lymphocytes. The B cells, they destroy bacteria and their toxins. They mature in the bone marrow and they produce antibodies that are specific to an antigen. So these bacteria have unique antigens on the surface of their cells, and the B cells learn their antigens and then produce antibodies specific to those antigens. The T cells, their favorite course on the menu are viruses, fungi, transplanted cells, which is not good if you need a new lung or a kidney or a liver. But good for us is cancer cells, and then some bacteria. T cells. T cells mature in the thymus. That's why they're called T cells. B cells in the bone marrow. That's why they're called B cells. And these two, the B and the T cells, work together to really combat infection and also to provide protection for or against future diseases. So these B cells actually form what are called memory cells, and those memory cells retain the information of the bacteria and the virus surface antigens, and that's why we can get vaccines, because we're getting the surface antigens without the actual live pathogen, and so our body learns those surface antigens. So when we get exposed to those surface antigens, then we don't get as sick as we would otherwise. All right, and then the last one with the cool name, these are natural killer cells. These are non-discriminate killers of infection, microbes, and certain spontaneously arising tumors. Thank goodness. Get rid of those guys. Okay. Then we have monocytes. These are 2 to 8% of the white blood cells. These are the largest. They're phagocytic. They're usually second to respond to an infection. However, when they do respond, it's usually in larger numbers and they're able to destroy more microbes. These monocytes, once they get in the presence of microbes, differentiate into macrophages. There's 2 types of macrophages. There's fixed macrophages and wandering macrophages. And their names kind of explain what they do. Fixed macrophages reside in a particular tissue and never leave. So when we talk about the respiratory system in the lungs, we'll talk about alveolar macrophages. When we talk about the lymphatic system, we'll talk about the stellate reticuloendothelia, or Kuffner cells. So we also have the wandering macrophages. Those will roam tissues and gather at sites of infection or inflammation. Okay, so what happens in death? Most of our white blood cells, honestly, are destroyed in the line of fire. They're destroyed while breaking down bacteria. Either they're destroyed by the bacteria and viruses themselves, or as they're battling it out with the bacteria and the viruses, they get engulfed by macrophages and then broken apart and recycled. T and B cells, there's always an exception. T and B cells can stay in the body for months or years as protection from future pathogens. Their memory. Again, this is why vaccines work, because they remember what we've been exposed to. If you're sick, a physician will order what's called a differential white blood cell count. That can tell the physician what type of pathogen you're fighting, or if something has suppressed the immune system. And both of that is very important information. Essentially what this does, the differential white blood cell count counts the number of each type of white blood cell. Okay, so let's try a practice question. So answer the following questions based on what you have just learned about leukocytes. Ben's differential white blood cell count shows a higher percentage of monocytes. What pathogen do you think he might be fighting? That's question 1. Question 2 is conversely, Maria's differential white blood cell count shows a very low percentage of basophils. What does this symptom suggest? So there's 2 questions there. Stop the video, practice those. And then, I say this in your green book, but just practice these. Answer the following questions based on what you have just learned about leukocytes.